In this video, we're going to talk about the biological aspects of dream high. Systems biology is based on the understanding that the whole is greater than the sum of the parts. In this course, we will explore how the knowledge of 1. Genetic makeup, 2. Gene expression, and 3. Clinical information from many cancer patients can inform the diagnosis and treatment of breast cancer. What is gene expression? In a cell, a gene is expressed when information is taken from the gene and used to create a protein. DNA transcription produces RNA, then RNA translation makes proteins. A cell expresses only a selection of the genes it contains at any one time. By controlling which genes are expressed, the cell controls its size, shape, and functions. Proteins are the workhorse molecules of life, taking part in essentially every structure and activity of life. To perform their functions, proteins communicate in a network, comparable to how humans behave in social media networks. In cancer and other diseases, there are changes in proteins that lead to miscommunications. These changes can be detected indirectly by measuring the levels of mRNA molecules that code for the proteins. Cells from healthy breast tissue and tumors have different characteristics and different gene expression profiles. Once a genome has been sequenced, we know what potential a cell has, what characteristics and functions it might exhibit based on the genes it contains. However, sequencing the genome does not tell us which genes a cell is expressing. Knowing which genes are expressed, the cell's gene expression profile, gives us clues about the cell's functions. If a gene is being used to make mRNA, it is considered on. If it is not being used to make mRNA, it is considered off. In gene expression profiling experiments, measuring mRNA levels shows which genes have been transcribed to mRNA, and thus have the potential to be translated to protein. For each gene, the number of copies of its mRNA is measured. mRNA levels are compared between patients. Some experiments can detect mRNAs for the entire genome at once. Usually, the amount of mRNA is measured in 2 or more experimental conditions or tumor samples, with the goal of figuring out how gene expression changes across conditions. Measuring mRNA levels from the tumors of many different cancer patients allows researchers to compare the expression profiles of the tumors and to associate them with clinical information, such as the type of breast cancer. This is a heat map of mRNA levels for different genes, the rows, from different patients, the columns. Green denotes genes that are expressed lower than average, and red denotes genes expressed higher than average. In this course, we will learn about the biology, diagnosis, and treatment of breast cancer by analyzing gene expression data. Gene profiling can be used as a diagnostic tool. If cancerous cells express higher levels of certain genes, and these genes code for particular proteins, then these proteins may be involved in the cancer. If it is known that one of the proteins helps cancerous cells survive, then its presence in the gene expression profile may indicate how aggressive the cancer is. If a drug exists that blocks the function of this or another protein, then targeting the protein with a drug might treat the disease. Gene expression profiling can be a key diagnostic tool for people with this cancer. Watch the next video to learn more about the computational aspects of dream high.